Hey guys, my name is Shreyas and welcome to Simple Biology. In this video, I'm going to be wrapping up the endomembrane system and talk about all the rest of all the organelles which work in the endomembrane system. So we talked about the endoplasmic reticulum and how there's two different types of endoplasmic reticulum, the smooth ER and the rough ER. And we already talked about the functions of the smooth ER, so let's go ahead and talk about the functions of the rough ER. So the rough ER actually secrete proteins made by ribosomes. And there's actually, I kind of briefly talked about this in our ribosome video, which you might want to go check out, but I'm going to be talking about that whole process, this whole process again in this video. Typically, these proteins which are made by the rough ER, which are going to be secreted, are glycoproteins. So, glycoproteins are actually proteins which have carbohydrates. Let me write this here. Glycoproteins are proteins which have a carbohydrate covalently bonded to a protein. Okay, so you have the polypeptide chain, and at the very end you have a carbohydrate. Okay, so hence the term glycoprotein. Okay, because glyco typically refers to something with carbohydrates. The rough ER is actually the membrane factory for the cell. It's what creates membranes. Okay, how does this happen? So there's actually a very extensive process here. So we have the rough ER right here. We already labeled what the rough ER was in this slide. So what will happen is that we know that the smooth ER is going to create phospholipids, right? So let's just say this is going to create phospholipids. And then we also have proteins which are made by ribosomes, just maybe free ribosomes in the cell. Okay, so if you don't know what a free ribosome is, go check out our ribosomes video. So we have proteins which are made by free ribosomes within the cell. So I'm just going to draw little dots like this to represent that. Okay, so what will happen? And then we also have proteins which are made by the rough ER. These little brown dots are all ribosomes. So those are going to create their own proteins, and I'm just going to draw that like this. Okay. So what will end up happening is that these phospholipids will go and they will attach to the membrane of the endoplasmic reticulum. These ribosomes, some of the ribosomes which are created by the free, some of the proteins which are created by the free ribosomes are going to go and they are going to enter the ER, okay, the rough ER, and then you already have these green proteins which are created by the bound ribosomes, and all this is going to form, and then from here you're going to eventually get a vesicle which forms. Let me see what color I can use. Okay, so you're going to get a vesicle which forms through, let me draw the vesicle right here. So the vesicle is going to have, which is going to have some of the phospholipids which were created by the smooth ER, and then it's going to have the green protein which are made by the bound ribosomes, and it's also going to have the orange proteins which are made by the free ribosomes, which kind of fused in with the rough ER. And this process happens through pinocytosis. When I talked about how this pinching happens, this whole process right here is basically pinocytosis. Okay, the term for that process is pinocytosis. So essentially, basically the same thing's going to happen where the membrane here is going to kind of pinch off, and then it's going to create this vesicle here. Okay, so from there, something is going to, this is called a transport vesicle, by the way. It's a special type of vesicle. It's called a transport vesicle. And that transport vesicle is going to go, and it's going to go and fuse with the next part of the endomembrane system, the Golgi apparatus. Okay, so the Golgi apparatus, what does it do? Okay, so it manages, it ships, and it sorts, okay, materials within the cell. Okay, so notice, manages, ships, and stores material in the cell. So the Golgi apparatus has a stack of flattened membrane sacs. Okay, as you can see here, these are all just sacs, flattened membrane sacs. Here we have 4 right here, and these are also just like in the ER we talked about the sacs being called cisternae. These are all cisternae as well. You have 4 cisternae right here. Okay, so what's going to happen is that, again, like I said, they function in protein sorting, packing, and shipping. And then before we talk about what we have the vesicle right here, which we formed. Don't forget about this vesicle here. I'm just going to draw it in here. Okay, and then... Okay, so we have that vesicle, which is formed by the rough ER. And then, before we talk about what happens to this vesicle, we need to make sure we understand this. There's Golgi apparatus. The Golgi apparatus has actually distinct sides. So each of these cisternae have distinct sides. We have the cis face, which is near the ER. The ER is going to be somewhere up in this region over here because you have the nuclear membrane here, and then we have the ER over here, and then right underneath that we have the Golgi apparatus right here. If you don't understand this, you can go look at a picture of a cell and you'll be able to see how everything kind of falls in one order. Okay, so the sides, the cisternae, the sides of the cisternae which face the ER are called the cis face. So these are all. So this is all the cis face. Whoops. So everything, all the sides of the cisternae which face the ER are called the cis face. Okay, and then the trans face, which is use green. The trans face is the sides which face the let's just use this. Okay, so the trans face is the sides which face near the cell membrane. The cell membrane is right here. So again, we have the nuclear envelope, then we have the ER, then we have the Golgi apparatus, and then at the very end you have the cell membrane. So the trans face, oh boy, trans face are these sides of the cisternae. Okay. Okay. So what's going to end up happening is that our vesicle, which we form here, I'm just going to go ahead and erase this. So our vesicle is going to come and it's going to fuse with the first cisterna. Cisterna is the singular word for cisternae. And it's going to come and fuse, and then there's going to be packaging and shipping. And it's going to come and it's going to fuse here, and then same thing. It's going to through parasitosis. Another vesicle is going to be formed, and then that vesicle is going to fuse with the second cisterna, and then it's going to enter this cisterna, and then there's going to be all sorts of processes which happen, and it's going to happen over and over again until finally we get our last vesicle right here. Okay. So through this process here of how this vesicle comes and fuses, the smooth ER actually creates polysaccharides. Okay, very long polysaccharides, which a cell might use. How does it do that? So remember that here I talked about how the rough ER secretes proteins, and typically these proteins are glycoproteins, and the glycoproteins are carbohydrates which are bonded to proteins. So try to use some thinking here. This is intuition. So if you have these carbohydrates bonded to proteins and then they enter the Golgi apparatus, what's going to happen is that the Golgi apparatus is going to pinch off the carbohydrates parts, and then those carbohydrates, which are monomers. Ooh, see an application of this. Okay, if you have no idea what I'm talking about, go watch the biomolecules and you'll understand. So these monomers, the monosaccharides, which are bonded to the glycoproteins, are going to be pinched off, and they're going to combine, and they're going to form polysaccharides, which the cell can use, and we're just left with the protein. Okay, so that can happen. Sometimes the glycoprotein will not be converted into just a protein. It can just stay as the glycoprotein. It all depends on what's going on in these cisternae. Okay, and then we finally reach this final vesicle over here, and this final vesicle is going to have little tags. Okay, kind of like whenever you ship mail, you have the address on the top. right? So you know where the mail is going to be shipped. In the same sense, the Golgi apparatus have these identification tags, and these can be things like are typically functional groups, um, things like a phosphate group. Like a phosphate group can be one of these tags. And basically, these tags, these tags are going to go and they're going to tell the vesicle what to do when it reaches the cell membrane. Again, remember that the cell membrane is lying right outside here, and the vesicle needs to go and fuse with the cell membrane. So these tags are going to actually help in identifying where exactly on the cell membrane that the vesicle goes. Okay, this whole process of how the vesicle comes and it joins and it goes back and it joins and it goes back like this. This whole process is called the cisternal uh, maturation model. Okay, so this is all just a model. Scientists don't even know if this whole process is even true. It probably is, but it's just a model and it's just a theory. But you need to know it. Okay. So now let's talk about lysosomes. Now, an important thing about lysosomes is that they're only found in animal cells. They're only found in animal cells. You never see them in plant cells. Okay. Now, they're structurally similar to vesicles. Okay. They're structurally similar to vesicles because they are formed just like how vesicles are. So as you see here, we saw the vesicles, a vesicle being formed from the rough ER, and then that vesicle travels through the Golgi apparatus, and then we end up with a transport vesicle right here, right? And that transport vesicle is going to go and fuse with the cell membrane. What can happen is that you can also get another vesicle, and instead of that vesicle going and fusing with the cell membrane, that vesicle is going to go and do other things instead of fusing with the cell membrane. And that, again, all depends on the tags. Okay, the tags tell what it's going to do. So this vesicle can go and form a lysosome. Okay, lysosomes have an acidic environment. Okay, remember, acidic environment means the pH is less than 7, and you have an excess amount of hydrogen ions, more hydrogen ions than hydroxide ions. If you have no idea what I'm talking about, you need to go watch the introductory to chemistry unit, because I'm telling you, everything in biology just builds up and builds up and builds up, and you're going to start developing holes in your knowledge and not understand what I'm talking about. And you can already see this if you don't know what the heck an acidic environment is. Okay, so anyways, so they have an acidic environment here. This is the lysosome here, by the way, and they also store digestive enzymes. They store digestive enzymes, which is critical. Okay, so lysosomes can what they do is they fuse with other things. It doesn't necessarily have to be with vesicles, but they fuse with other things and they break down their contents. Okay, so lysosomes can basically do fuse with 3 different things. They can fuse with food vacuoles. Okay, so what would this be? So imagine you have a cell, and then you have some food. Let's just say we have some orange food like this. So the orange food is going to approach the cell, and then it's going to eventually kind of push into the cell like this. And it's kind of like the opposite of pinocytosis. This is called phagocytosis, which we'll be learning about later. And then eventually you get this, and then you have the food inside. This is called a vacuole right here, which we'll be looking at in just a second. But what will happen is that the lysosome is going to come around, and it's going to, with its digestive enzymes, let's just draw that in here. Its digestive enzymes, it's going to come and it's going to fuse with this food vacuole and digest all that food. So, maybe you have like a very, very extensive polysaccharide or something like that, and that needs to be broken down before it goes and it's actually useful to the cells. So it can digest food vacuoles. It can also take old cell parts and old cell organelles and break them down, and then take those and the cell can take those components and turn it into new organelles. Okay, and it can turn it into new organelles, and this whole process is called called autophagy. Okay, I have an example here. We have a lysosome right here, which I've already drawn an arrow for, and then we have this is a chloroplast. This is a chloroplast, and the chloroplast is, as you can see, what will happen is that you're going to see kind of this membrane which forms around the chloroplast, and then the lysosome is going to go fuse in, and it's going to release all its contents, its enzymes, its digestive enzymes. It's going to go and release all these digestive enzymes. They're going to break down the chloroplast and turn it into smaller biomolecules which can be used to form different organelles. And finally, this is what we are probably thinking about. The lysosomes can kill pathogens, which is probably the most important thing. Whenever you get a pathogen which enters the cell, the lysosome is going to use its digestive enzymes to kill it. Okay. Finally, let's talk about vacuoles. Okay. So, actually, I'm going to leave this for a final third video because this video is also getting long, and vacuoles have a lot to say about them. So go stay tuned and check out our final endomembrane video where I'll be talking about vacuoles. That's it for now. The endomembrane system is as simple as that.